Adenosine triphosphate is the molecule that we're seeing in this picture here. And this is a molecule that is very important in the cell for a variety of reactions. And specifically, this is important and it's very useful for endergonic reactions. And the reason for that is that this is a molecule that can hold on to or store a very useful amount of energy inside of it. ATP is used as a short-term energy storage molecule in the cell. And so since we're going to be storing energy in this molecule short term, it is going to be very useful if we just need to run a single endergonic reaction, or if we need to run an active transport, just a variety of things that we might have going on. If we look at the molecule itself, the part that I want to focus on is the part shown in blue, and that's because that's where the phosphate groups are. And the phosphates are the part of the molecule that really holds the vast majority of the energy. So these three phosphates, if you look at them closely, if you'll notice here that they do have these negative charges on them. And so in this case, you've got all these negative charges, and like charges repel each other. But when we have the three phosphate groups together like that, it's like a compressed spring. So those negative phosphate groups have been shoved very close together, and that makes for a very highly unstable portion in the molecule. And remember, as we've been saying, the more unstable something is, the more energy it contains, the more potential energy it contains. So that little phosphate tail is what we call it, that has the three phosphates. That is a very high energy portion of the ATP molecule. And that's where the energy that's stored that we're going to utilize for cell reactions is located. Now, specifically, where it's located at would be actually between the phosphate groups. So these little squiggly lines, they are squiggly, representing these are very unstable bonds. And again, since they are unstable bonds, they're going to actually hold a lot of energy inside of them. So they're shown as squiggly lines because they're going to break very easily. And that's another reason why this is a short-term energy storage molecule. Since the bonds are so unstable, they're not going to stay put for long periods of time. So if the cell does need to do long-term energy storage, it has other resources for that. And that would be something like a fat molecule. Here we have a very simplified drawing of ATP. When ATP is going to be utilized in the cell as an energy source, the first thing that's going to happen is that ATP is going to be hydrolyzed. And what that means is that we're going to get a hydrolysis reaction. So for our hydrolysis reaction, ATP is going to be cut with water. It will be that phosphate bond between the second and the third phosphate. So I'm going to label this as my second phosphate and my third, and this would obviously be the first. So we're going to cut it right there. And the end result of that is that we're going to get what we now call a DP. And a DP is going to really be what was left. So we have this situation where we have two phosphate groups still attached. And then we also have the phosphate that we cut off, which we call inorganic phosphate. And so it's designated with PI. Along with that, we have energy that was released. And that's really the portion of this reaction that we're interested in. So this reaction, as we see it here, is going to be an exergonic reaction. Exergonic reactions are useful because they release energy. And the cell needs energy to run endergonic reactions, or anabolic reactions, we could call them as well. So this is called ATP hydrolysis, as we see it right here. Now, ATP is going to continuously be broken down to use for reactions inside of the cell. So we certainly want to have a mechanism to actually put ATP back together again, or to recycle our ATP components. So if we look at this another way, ATP is going to get broken down into ADP and phosphate, inorganic phosphate. The reason for that is because when we do that, energy is going to come out. And that energy is useful to run endergonic reactions. Okay, so we would utilize that energy somewhere in the cell for active transport of something, for just a chemical reaction, something anabolic, like if we're going to build a protein, variety of things we can do with that. Now on the other side, we want to be able to complete this circle, but when we are completing the circle, that would actually require the energy come in. So we have to have a source of that energy. And so where we're going to get that energy from, that would be essentially food. So when we eat food, we do that because we need energy, and you've probably used that terminology before, but food molecules will themselves be broken down to get some energy out. And once the cell gets that energy out, it's going to actually rebuild ATP molecules with that. So when we do that, this would be like compressing, if you will, the spring. And by spring, I'm talking about the phosphate tail. So we're shoving those 3 negative phosphate groups very close together, and they don't want to be like that. So since we are going to be making ATP much more unstable, we'd have to have energy to do that. On the other side, instead of compressing the tail, we're basically releasing the spring. And since we're releasing the spring, ATP would then become more stable, which means that energy is going to be released. So this is the cycle of ATP. Inside of the cell, this is going to be continuously happening. And just to put a few other terms on here, when we do perform ATP hydrolysis, which is the breaking down of ATP, that reaction right there is going to have a negative delta G. But when we're on the other side and we're actually going to build ATP, that reaction is going to have a positive delta G.